Okay, welcome back again. So after the presentation of Dr. Fatima about the plant-based meat, analogs, so I will start with my own presentation that is understanding the structure of meat analog products. That is a literature review we are doing with Dr. Fatima. She invited us to write a review paper. We collect some articles, and so I'm going to discuss these articles with you. But first of all, in the past presentation, Dr. Fatima told you about the availability of these plant-based products. So I will share with you some basic products that we have here in Mexico already. This is cheese. This is a mixture of cheese. I hope you can see. And this one are burgers, burger patties. So these burgers are based on lentils, barley, carrot, pumpkin, onion, beans, salt, and spices. So you can see the patties here. And also, of course, you know the almond milk, barley milk, coconut milk, and so on and so forth. So we already have.
We can.
Yes.
I can see the slides if you are changing.
Yeah. No, I'm not changing the slides. I was sharing in my camera. I was sharing this.
Okay. Yes, I cannot see your camera now. I can
Okay.
hear. Yes.
Okay. So we can. This is cheese for pizza. It melts, so it's very tasty. These are hamburgers, and of course we everybody know the almond milk, soy milk, and other plant-based milks. So it is a trend. It's a world trend now that is being already available for large consumers. So you heard the benefits of plant-based analogs. So it's environmental and healthy benefits together. So okay, these are some commercial brands that exist in the United States. I show you some of the brands that we have. This is interesting. You see, this is a plant-based meat that it's very similar as chicken. So Dr. Brishti explained how to reach this texture. No? So there are some show some detailed photograph reaching this meat texture. So they are as in material science, they have two different strategies. The bottom-up strategy that begins with structural elements, so directly the proteins. For example, they use microproteins. They do injected meat or cultured meat, and wet spinning and electron spinning. So they begin with small parts, the amino acid mixture or the protein mixture, and they assemble the elements and get the fibrous structure. So there is also the top-down strategy that begins with a mixture of proteins and polysaccharides, and then they make an extrusion and compress and do some pressure and do some heating, and they finish with the texture of meat-like. No. So this is how the final products look like. You see, this is laboratory extruded and shear cell processed meat. So it has the typical fibrous structure of the chicken meat, the pork meat, the beef meat. But there is apart from the texture, about from the looking side, because you look this and you said, okay, this is meat, but because if you have tasted, for example, soy texturized soy meat, it doesn't have any flavor. It's just you have to put all the spices there. But if you want to have a product such as this, the burgers I just show you, they have to taste as meat. They have to feel. You have to bite it, and you have to feel the mushy texture, and you have to feel the flavor coming from the meat. So it is it is very important, the processing parts, no? So the fat that is mixed together with the vegetable protein and the additives that give flavor, and the moisture that is a part of the cooking process, because you put the meat onto the pan and it loses some water and it loses some fat, but it keeps some fat and some moisture inside, so it can be more tasty. They depend on the microstructure. Also, things like the processing. For example, you have the texturized meat like this one, like it's like a kind of a powder. When you form the patties, when you form the sausages, they have to be they have to preserve the properties. Also, after cooking, they have to be attractive to the consumer. No, you don't want to eat something like looks like carton, or like looks like paper and tastes like paper. No, and also the digestibility and nutritional aspects. No, Dr. Brishti just speak about the balance between the amino acids, the fat, and the moisture within the plant-based meat analogs. No, so to know all this, to know how the structure is developed and keeps during the different parts of processing and digestion and so on, there are several these characterization methods, mainly microscopy and spectroscopy techniques. But first of all, how the meat looks like, because we have discussed meat-like analogs, plant-based meat analogs, but how the meat looks like, how it is the meat inside. No, so this is a large-scale scheme of the meat cut. So you have, this is the macro structure. So you see these small structures, no, that as you approach to the micro and nano structure, these fibers, these meat fibers, are made of two different proteins that are actin and myosin, that impart the flexibility and toughness of the meat fiber. So inside any typical meat, you have these fibers. You also have fat, an amount of fat that depends on the type of meat. You have the sarcoplasm, that is a liquid that involves, that is around all the meat, and the connective tissue, that are the tendons, the nerves, and so on and so forth, that are made of collagen and elastin and so on. So this is the structure we want to mimic, no, to have a product that really looks and tastes like real meat. No, so in the natural meat products, there are two very important things for the processing. They have to have cohesion. Cohesion means it has to hold together both the meat and the fat and the water. They have and the flavors. They have to hold together, and they have to have the texture, no? So how it feels in the tongue, how it looks when you cut it, how it looks when you how it behaves when you process it. So these two things depend very strongly on the exudate formation and the comminution. The exudate is a solution of sarcoplasm and myofibril proteins. This is a natural thing that comes from the meat. This exudate is formed when the meat is processed by mechanical action, such as massaging and tumbling. For example, if you put you put the meat and you cut it, and you how is said in Spanish, in English? You put it in the blender, for example, or in the mill when you have some mindset meat. So some exudate is obtained. You have seen it's not blood. It's something called exudated that is formed, as I said, from sarcoplasm and proteins. So this exudate comes from the meat when you damage the connective tissue. So this scheme shows how the texture changes as much as you do a greater comminution, you do more mechanical action, and you get more binding between the components of the meat. For example, you have the cut. This is called steak. This is the big piece. It doesn't have any processing, any comminution, not cut, not anything. So you have the cohesion is complete. So when you cook it, it keeps the shape. It shrinks a bit, but it keeps the shape. So it has this fibrous texture. So you fight against the cut. No, it has you see the fibers. No, but when you begin to do comminution and cutting and processing, more mechanical action of the blades, for example, and of the meal, you begin to get this the meat that can use it for burgers or for sausages, for example. No, you see, when you begin this processing, when you increase this processing, the meat is very cohesive, So the sausage, you can just snap the sausage into parts. No, you can break the sausage with your hands. You cannot do that with the steak. No, the steak cannot be broken. It is fibrous. But after the processing, the meat begins very cohesive, but it doesn't have any fibrous texture. No? The sausages doesn't have any fibers inside. So you can alter the texture and all the protein and all the properties of the meat after doing this kind of processing. So this is the type of things that we want to mimic when producing plant-based meat analogs. So, for example, there is a technique that is called last scanning microscope. This is the principle of operation. You have a laser here that is reflected onto a mirror. So it comes onto the detector. So the thing here is that why it is called conform. The focus plane can be changed. So you can move a movement in the vertical, we can say, the vertical plane. So you see, you can do cuts. You can do vertical cuts of your sample. The question here is, you use some fluorophores that absorb differentially on the different structures of the meat, of the cells and the fibers, etc. So they impart different colors. For example, here we have two confocal micrographs where you can see, this is the left one is beef and the right one is plant-based beef, and they are observing the lipid red color lipids and a red color absorbed on the proteins. So when you do a mixed image, you can do the separate images and do the combined image. You can see how the lipids and proteins are combined in the natural beef in the mouth. So this is really wanted to simulate all the digestion processing, all the digestion process. So when you eat the meat, how the lipids and proteins are distributed on the mouth, and then how are distributed in the stomach. So you can see they are dispersed, completely dispersed, and then how they look in the small intestine. See, you can see that there is almost any presence of protein or fat inside the small intestine. So if we go to the plant-based beef, you see that the lipids and proteins are more distributed in the space. No? So they are more small structures, because the plant-based beef, as we saw, it is made by processing, no? by extrusion or by shear cell-based technology. So the lipids and proteins are very well distributed, and in the stomach, some non-separated parts of the lipids and the proteins, and after the digestion in the small intestine, you can see these red spots in the intestine that indicate that not all the lipids are digested as there were in. That's why, because the plant-based beef, as we discussed it before, they contain a lot of fiber. So it's known that the fiber binds the lipids and avoids that the lipids incorporate to the bloodstream. So they are eliminated in the intestine. So this is a very good result, because it indicates that the lipids have to be incorporated into the meat to give texture and to give flavor and to keep the flavors together. But they are not digested as on the plant, the beef, natural beef. Of course, there are. We want to see the texture. We want to see how the plant-based beefs look after the processing, or during the processing, or during the digestion. So, as scanning electron microscopy and transmission electron microscopy are very good tools to observe the microstructure. No, in the past talk, we observed how these small sacs appear that can hold both the water, the lipids, and the flavor in the materials with different processing conditions. And for example, this is a TEN micrograph, transmission electron micrograph, on natural beef. You can clearly see how the microfibrils are aligned, no, in this direction. So, or also in this direction. There are two directions here, and this one and this one. So this is again the structure that is interesting to mimic in plant-based analogs. Another interesting tool that is useful for characterizing plant-based meat analogs is atomic force microscopy. For example, this is the scheme of the technique. As we know, we have a cantilever. At the end of the cantilever, we have a tip that touches the surface of the material, and a topographical map is made on the reflection of this laser on a photodetector. So after moving the piezoelectric scanner, you can make a two-dimensional and three-dimensional map. For example, this has been very used for characterizing cellular membranes. In this scheme, in this upper left scheme, we have the vision of intermembrane proteins. So many proteins are inside the cellular membrane, so they can be imaged both by contact AFM or by tapping AFM. These are two examples on characterization of different plant-based analogs. For example, this bottom left are soy-based proteins after different processing times. So you can see how the soybean protein acquires a fibrillar structure here and here. And also, this is another atomic force microscopy study on sein lecithin. Sein is the protein that comes from the corn, from maize, and lecithin is a known emulsifier. So emulsifiers are very used in food technology, hold together and the fat and the flavors. So the interaction between the proteins and the emulsifier is very important to study in order to make good formulations to good texture and good flavor, good taste. No? So you can see how the structure evolves after changing the proportions between sein and lecithin in different conditions. See? Another interesting technique that allows us to see the volume structure of the plant-based meats is X-ray tomography. You know, X-ray tomography is used in medicine to observe the living tissues, the bones that can be, you can have a cut, a vertical cut of the bone. So X-ray tomography uses exactly the same principle. There is a scanning, a vertical scanning of the sample, so you can see this is the principle here, and you can reconstruct a 3D map of how that includes not also the shapes but the porosity, the void volume inside the structures. No, so you can see how much pores, and you can see the pore size, and you can see the volume that is inside the sample. No, this is in an inorganic material, and this is in a plant-based analog. Where you can see this is the structure. You can see in orthogonal slices. That means in the same plane of measure, how the porosity develops with processing and with formulation conditions. So these voids are important because they impart both elasticity, and they can hold also the additives of the plant-based meat, such as humidity, fat, and flavors, and etc. Another very useful technique that is for identified, for example, the profile, because we discussed that it is very important to have a balanced amino acid profile in plant-based meat analogs, because a single plant doesn't give all the amino acids that the human body requires. So you have to have a mixture of different plants to have a correct balance of these amino acids. So the technique of excellence to see these amino acids in plant-based materials is nuclear magnetic resonance, that it bases on how the proton of inside the materials moves or resonates when it's subjected to a very intense magnetic field. So with this technique, you can see here, this is the entire amino acid profile of a sample, and also you can identify, for example, the saccharides, the monosaccharides and polysaccharides inside, and also you can identify the fatty components, the fatty acids that are inside a plant-based meat analog. So these are typical nuclear magnetic resonance graphs. So you can see the different the shifts of the proton. So each peak is related with a type of proton in a part of the molecule. So you can do a qualitative analysis of these components in the plant-based meat analogs. A special mention that I wanted to stress out in this presentation is the use of synchrotron-based methods. The synchrotron-based methods have the same principle of the regular methods. For example, Xray diffraction, XPS, etc. The difference, for example, an Xray tube, a bolometer, or incandescent source, for example, but it comes from a synchrotron. So a synchrotron is a big device that has that first it has this device that it's called electron gun that generates electrons, and they are accelerated these electrons are generated in a similar way as in an electron microscope. They have a lanthanum boride ceramic that offer a voltage in electrons. Electrons are accelerated in this device, linear electron, up to 100 mega electron volts. So these electrons are first injected in this so-called booster ring. So this booster ring is like a blender that makes stirrs, accelerate the electrons up to some hundreds of mega electron volts, and there they are injected into this second ring that is called the storage ring. So the electrons in the storage ring are accelerated up to the light speed. The energy of these electrons can be around 3 giga electron volts. So after these electrons come out from this circular trajectory, like in this rays, this tangent rays that come from the storage ring, these are attracted by the so-called insertion devices. That is a group of magnets and electromagnetic fields that bend the electron beam inside this chamber. No, so once the electrons are extracted, they become radiation. They can become from X-ray to infrared radiation, depending on the type of the extraction device. So this is the chamber, the optic. So you can collimate, you can concentrate, you can do all the processing here. And here in the experimental hutch, you put your spectrometer, and it is controlled from a workstation. So the advantages of the synchrotron-based methods is the light is very much brilliant. So they have very high orders of magnitude of photons above a regular source, and these photons come very, very well collimated, so they do not disperse as in a regular source. They don't open as they propagate. They are very collimated, and also they can be polarized. Yes, so the measurements done in synchrotron-based methods are more precise and have the possibility to observe with more detail all the things that you can that you want to study. No? So one of the techniques that is very interesting for studying plant-based analogs is the Fourier infrared spectroscopy. The Fourier infrared spectroscopy in a synchrotron, the light comes from the synchrotron, as I mentioned. So the light comes in the mirrors. They have a beam splitter here, and then they go into the sample. No, in fact, this part of the device is a commercial infrared spectroscope as the one we have in CICATA. We could take our infrared spectroscope and put it in the synchrotron line and just take out our source and put the beam there. And so you can use the light coming from the beam with our own spectroscope. So we have the sample and the detector and so on and so forth. So one of the most interesting things to observe in the plant-based meat analogs is the structure of the proteins. A protein is an entity that has some nanometers, but it's very packed. It's very packed and has different internal structures such as alpha helix. So this alpha helix is made of different amino acids, and they have also these so-called antiparallel beta sheets, these yellow ones. Antiparallel means that one's coming in this direction and the other is in another direction, and this is the other one called beta strand. So these structures are very important to understand how the amino acids are bonded inside the proteins in the plant-based meat analogs. So you can see the wave number region. This is the so-called protein region. It's around 1600 and 1800 centimeters power minus 1. So here you can observe the proteins, the lipids, the nucleic acids, and carbohydrates. And this is a derivative plot. These are synchrotron-based FTR measurements, and you can really discriminate the bands coming from this particular structure. No? For example, these bands could be associated to the alpha helix and those to the antiparallel beta sheet, etc. So it is you have a better resolution power using the synchrotron light compared with the infrared light, for example. Another technique that is commonly used for studying materials is X-ray fluorescence. We can this is based on illuminating the sample with a monochromatic X-ray beam, and this monochromatic X-ray beam makes the atoms inside the material to emit X-rays of a very particular energy. So they are the digital fingerprint of the element inside the material. For example, we have this plant-based meat analog sample, and we have the profile of phosphorus, calcium, potassium, iron, zinc, and copper inside the material with a very good resolution power. For example, this is a membrane, and you can see how the different elements are outside in the membrane, and how these other elements are into the membrane. So this is made for elemental analysis, not for structural analysis, but as we discussed earlier, it is very important to keep a very good balance on the minerals that are inside the plant-based meat analog food products, so as iron, as zinc, that impart important nutritional properties. This is an example of synchrotron-based small angle X-ray scattering and wide angle X-ray scattering. When we talk about small angle X-ray scattering, we are talking about the diffraction that comes from the sample at diffraction angles below 5 degrees. So we are used to work in our laboratories to wide angle X-ray scattering or X-ray diffraction that comes, for example, from 5 degrees up to 30, 40, 70 degrees that are useful to observe inorganic structures. Because as you remember, the plane distance is inverse to the diffraction degree. So smaller smaller lattice constants are observed at bigger diffraction degrees, for example, in inorganic cells, inorganic crystals. But in the case of macromolecules, such as proteins, such as carbohydrates, the molecules are very big, so the lattice parameter is very big. So the diffraction, it has to be observed at very low angles, so at angles below 5 degrees. So the Sachs region is from the left to this one. Yes. So this is our typical SAX scattering diffractograms. So they are plotted usually in terms of nanometers -1. No? So here in analyzing this, you can observe how the structure of amylopectin, that is one of the polymers into starch, is changed when you add water. No? So adding water disrupts the structure of amylopectin, and after becoming a linear change, they became these circular crystallites. Yes, so the SAX is very good to observe macromolecular entities, and this is an example of in situ diffraction, wide-angle X-ray diffraction. These persons have this. This is a shear cell device. So while processing the plant-based meat analog in this chamber at different temperatures, you can obtain the diffraction patterns, and you obtain this. For example, processing at increasing temperatures, you observe a disruption of the structure. The material became amorphous after the processing. But this can be observed in operando, so during the processing. There is another technique that is very common to study proteins in plant-based meat analogs, that is circular dichroism. Circular dichroism is based on the differential light absorption when you change the polarization angle. You know that electromagnetic waves can have planar or circular polarization. So when you put a circular polarizer between the incident beam and the sample, you change the polarization state, and you observe differences in the spectra when you have different structures. No? For example, in these cases, you have this is the ultraviolet range of electromagnetic light. This is made in the synchrotron, by the way. In the synchrotron radiation, in the synchrotron sources, you can see beyond 200 nanometers. The regular sources, in the laboratory sources, are work from 200 nanometers up to 1100 nanometers. So you lose this important part of the ultraviolet range that is below 200 nanometers, as you can see here. For example, 200 nanometers is here, and you have very wide responses below this. So this is made in a synchrotron light source. So, for example, you have here different protein structures that can be readily identified by circular dichroism. You see that the response is very different from all the different kinds of proteins. So after you study, for example, here you have different protein sources. We just discussed the different plants that are used as protein sources in plant-based materials, such as oat, soybean, potato, whey, mung bean, and they have the different contents of fibril and non-fibril structures, as you can observe here in the differential circular dichroism response. And this is all from the characterization techniques we have reviewed in this paper. So I want to thank you all for your attention. This is our research group. Have Mrs. Sammy Chong in the masters. Also Mrs. Fer Gonzalez is in the masters, as well as Sergio. The PhD students are Fernando, Marcela, and Rafael. Yana Silan is alumni. He's already a PhD from us, and Dr. Eric Alarcón, that is a collaborator from us. So thank you very much for your attention. I will be glad to answer your questions.